of the biology of the eye. So we talk about what happens to the eye lens and to the cornea as a function of time, what the basic photochemistry and the biology is. What has happened in the field of eye biology has not been just a contribution of molecular biology, genetics, or cell biology alone. It has been a great collaboration where deep insights have come not just from these areas of natural science, but also from physical sciences, engineering sciences, and material sciences. And I thought I'll talk about some of that today, where physical science catches the eye. What you see there is a beautiful picture by Salvador Dali, and you notice he thinks of the eye as a jewel. Last time I talked about it as an eye, an organ whom age withers and custom fades. It does indeed happen as a function of age. The function of the eye does keep on deteriorating, and we need to find ways to address this problem. There are various ways of looking at that. Of the organs in the body, one that is particularly easy, so to speak, to operate with is the eye, because it's isolated, put in a socket, and thus, in terms of interaction with the other parts of the body, the only kind of an interaction it has is by and large through the circulatory system. But other than that, you can treat the eye as pretty much of an adiabatic. What I'll talk about today is less on biology and concentrate more on what polymer chemistry, material science, and even physical sciences like optics and engineering have been contributing. The focus is in particular areas such as presbyopia. If I look at the audience here, many of you are already wearing near vision glasses. As a function of age, near vision seems to be a problem. There is also the other situation, along with that, myopia or short-sightedness of the eye also increases as a function of age. Increasingly also, the globe is going through an epidemic, a pandemic of diabetes. And that affects the eye directly because of vasculature and so on in the retina. And we can wonder about what today appear to be interesting problems. You notice essentially what we have is an optic system here, where we have several refractive surfaces. The very first one is the cornea. That's up there. Pretty much almost 80% of all the refraction actually happens right there. And then you go to the aqueous humor and you have the lens which further focuses it on the fovea over there. We will not worry at the moment about a slight departure between the visual axis and the optic axis. But we will worry about the refractive indices. You notice pretty much the refractive index for the cornea is remarkably close to that of water. Therein, of course, is also an interesting, both a problem and also a solution. On the other hand, if you were to go to the lens, which is actually hung by the ciliary muscles, the cornea is not as flexible as you would like, whereas the lens happens to be sufficiently plastic, elastic, that you can actually change the focal length by changing the refractive index, by pushing or dilating the lens, which is very, very nice. And that's what is referred to as accommodation. We talk about accommodation here. You notice because of the ciliary muscles over there, you can actually make it oblate or you can make it more spheroidal. And that means you can relax it, the refractive index, then the focal length actually increases, and as you push it, the focal length decreases. This is by and large. But this accommodation is a property not only of the ciliary muscle and their give, but it also the property of the lens, which tends to harden as a function of age. There is an age-dependent dehydration that happens in the lens, makes it harder and harder. Therefore, it becomes less and less elastic and plastic. And that, by and large, is a problem that many of us face here and therefore we had to change to near vision glasses. And hence the idea of bifocal lenses. Of all people, it was actually Benjamin Franklin who actually invented the idea of bifocals. So rather than take one pair of glasses for distant vision, change it into another one for near vision, what if I were to simply put them both together? That's the first technology we had. Interestingly, this has just not moved beyond 1784. Technology-wise, this is by and large what we even have today. And as he points out, by this means, as I wear my spectacles constantly, I have only to move my eyes up and down, which is precisely what people do. The only technology that has now happened since Benjamin Franklin's time is you no longer have to have that particular break between the top and the bottom lens. You can actually have a progressive. I think many of us actually also wear progressive lenses at that time. So you don't have to be Benjamin Franklin's. Keep on doing up and down. And of course, I'm just pointing out in the footnote here that he not only invented the bifocal, but he also did a variety of other remarkably interesting things. The question we're really asking is, therefore, what can be done to rid of bifocals? Is it possible at all? What can science of today do to help? And can we get rid of near vision glasses completely? In other words, presbyopia, how do you address that problem using material science? There could be a possibility that I can remove my lens and put in a plastic intraocular lens. Generally, the plastic that is used is PMMA, polymethyl methacrylate, or hydroxyethyl polymers of various kinds and so on. It may even be possible, is it, to replace the lens simply by a polymer gel in a manner that essentially becomes something like a crystalline rich biological eye lens. And here polymer chemistry seems to have come into help. We have one set that have been actually put together by Bosch and Lone, which is called crystalline. And you notice this is exactly like the normal intraocular lens that one wears, with the haptics over here and there. And this must give, just like here is a single optic. Still the lens is only one in terms of optics. And what it does, if you put it in there, again, the ciliary muscles will do their trick and it moves back and forth in order for you to give. And this movement back and forth is really done by the eye itself, by our own ciliary muscles. In other words, you don't have to do a Franklin of going up and down. But as you see, just like in a normal eye, where the lens is perfectly all right, near vision and distant vision are both accommodated quite easily just by moving the ciliary muscles back and forth. So this is one of the substitutes that has come about in the last 2-3 years or so. And the material in there essentially is a polymer. And the 12-month phase 2 clinical trial was done in our laboratory, and we find that after 1 year follow-up, at best you could do is accommodation in our diopter is no more than plus minus 1. That's not very good. So you need to do something better than that. How about a dual optic accommodating intraocular lens? What you do here, very interesting contraption here, where you put in 2 lenses rather than 1. That happens to be a positive power, and this happens to be a low negative power. So this is in the back, and that's in the front. And you actually put it in there, and we have tried this in our institute. We find that you still use the same natural forces of ciliary processes. And the change, the optic to optic separation, that to that separation, okay, is essentially handled by the ciliary muscles themselves. Here is one such implantation. You can see that. Dr. Samwan, my colleague, has actually been attempting to do this. You'll notice there is that, and he puts it in there. And once it is done in there, it essentially lets the natural ciliary muscles do the trick. How good is it? Once after it, we have done this on several patients and tried to look at how good this technology is going to be to win over the accommodation problem. When you do that, the best corrected vision seems to be both in terms of the range and the mean, but you notice essentially what you have is I can now increase my diopter value of focal length and go all the way up to +3 diopters. By the way, a diopter is the inverse of the focal length written in meters, so that you get an idea of what the diopter is going to be. This is what we call the power. When you say I am -3, -4, and so you're talking about the diopter value. And here, you actually were able to add up to +3 diopters, but you still need more than that if possible. And thereby, the suggestion has been made that rather than use plastics at all, can we use good polymers? And what this smart lens does by Professor Fine, about 2-3 years ago, is that he will completely remove whatever is there in the lens. All the time you're not planning on the lens, remove the natural material of the lens and put in there the polymer rod, and put the rod over there that is put in there. And as you get into that, you have to cut the top part of the capsule that surrounds the lens, introduce in there. So there is an anterior capsulectomy that has happened in here. And this fills completely. And because of the natural hydration processes that happen in there, you can actually have a soft and compressible disc that happens in there. That's how you inject it. And this is referred to as a phaco-ersatz theory. Notice what has happened in there. You put in that little rod and it completely covers it. And that happens to be the ciliary muscles in there. That's the iris. This is by and large the procedure, but you notice again here, what is being done is you take a little bit of that polymer rod, put it in there, and it completely covers. So that what we now have is essentially a polymer gel that seems to accommodate much better. Material science and polymer science seems to be of value in here. And yet there are unresolved problems. Achieving emetropia, normal vision in relaxed state is important, because once I put in these, I still have to have the difficulty. Optics must help in this one. What would be the ideal polymer with optimum accommodation and yet biocompatibility, so there is no inflammation that happens to the rest of the eye and so on? And the surgical technique for the hard cataract is going to be somewhat difficult. There may also be rubbing between the polymer and the back of the capsule, leading to what people refer to as posterior capsular opacification. So can chemistry, physics and material science help here? That's really the question that we're asking. We still do not have a good answer for that. And there is a huge demand for this because the number of people who would like this. That's what I'm pointing out here. Why is this obsession with both myopia and presbyopia? Because they're endemic in many parts of Asia. It's amazing. 90% of the youngsters, school-going children in Singapore and Taiwan become short-sighted, myopic, before they reach class 10. The epidemiology suggests that 32% of primary school children and 83% of university students in China, particularly in urban China, have impaired vision. We don't have such an estimate right now in India, but our own study in Andhra Pradesh suggests at least 0.17% of the children, to the extent Andhra Pradesh represents, with the 70 million, represents India with the 1 billion and so on. One could suggest out of these 0.17%, so 1/3 of them are really due to defective vision. And you talk about blindness, you really talk about legal blindness, and not vision completely lost. The blindness here is talk about what I see at 60 feet. This child is not able to see until you get into 6 feet or so. So we're really talking about close to 1.3 million children being myopic. Why is this? It's not just genetics. Genetics might account for a few, and there is also multiple genes involved in that. It is not a monogenic disorder, but it seems more and more possible that mere work, getting work with textbooks, with computers, whatever, enhances myopia. That's an interesting issue. People have studied, for example, Eskimo children, those who go to school, those who do not go to school. People have studied also in northern Mongolia, again, children who do near work children who do not do such near work, and there's clearly this association between near work and increased myopia. So we need to worry about that. In presbyopia, which is the age-dependent hardening of the lens, and therefore you end up getting near vision. Over 77 million in India are over 60 years of age. And that number is going to be doubled in about 20 years or so. But what is happening is when you look at statistics in India, presbyopia hits people already by the time they are 40. Our analysis suggests that 55% of the people studied age group, we notice 47%. In Andhra Pradesh, are presbyopia, and they need at least one diopter correction. Females have a slightly higher odds than males. That again might point to environmental factors in here. The odds ratio there you notice is 1.4. Rural residents 1.5. We don't completely understand. If you are myopic, you also turn out to be near vision and hyperopia. And translated to whole of India, this becomes significant. Therefore, just giving glasses will not do. We need to have newer technologies that come. That's what I thought we'll talk about. One way of approaching the problem has been you can correct vision by either wearing glasses or by putting contact lenses. What about a contact lens is used every night, you remove it. But today, there are even contact lenses that are possible that you can use for months. You can sleep with it, you can go to swing with it, and so on. But yet, it is still essentially a removable one. What if you want to simply stitch it or put an onlay in which what you do is you slice in 2-3 institutions, including ours. You flatten the cornea by actually taking one, I'll show you the video of that. You flatten it, you slice it up a little bit like an onion skin one, and you insert a thin refractive plastic lens and correct the refractive error. The lens stays there, and you can actually just about see where the arrow is. The lens, after it has been inlaid over there, stays over there, and 12 to 13 months after, there is no contraindication at all. The other way of doing that is not just go to the epithelium, but actually put one right in the stroma, which is the difference between an inlay and an onlay essentially is where you put it. Here is how the technology actually is done. By and large, this has come largely because of a physicist, a physical chemist, Dr. R. Srinivasan, who used to work at that time at the IBM laboratories, who used laser technology at that time to sculpt the cornea. But you can do exactly the same thing here. Notice you flatten it by using this little instrument over there. And you can also slice a very thin layer, approximately about half a micron or so thin. And you notice, now you can insert the lens over there, the contact lens over there. That is the square part that has actually been opened up, and you put in the lens over there. And this stays. Now, how good this is going to be, we still do not know. We have done work on this to try and find out whether the polymer is good, whether they stay, whether the inflammation and so on, we still do not know. Is that the best instrument to use? Again, we do not know. But here again, I think there is a lot of possibility for technologists. One other thing that has actually happened is myopia is not only affecting just that little part of the fovea or where we actually focus the light that comes in by first focusing from the cornea and then from the lens. It also turns out that it affects the peripheral retina, not just little spot over there, but right around that region over there. In other words, essentially what happens is you have, as myopia happens in here, you notice also once I correct it, I have corrected using a glass, but you notice at the retina it is still not exactly on the focus. The peripheral retina becomes increasingly slightly myopic. The glasses therefore that you use today are not good enough. The glasses you use today focus only on that little spot over there. Can we now have glasses that also look at focusing at the peripheral region? The periphery is not very large. It can't be over there and so on. It just essentially approximately about 2 to 3 millimeters on either side. So it is now possible. Can we now redesign spectacle lenses? Can we regrind lenses in a fashion that they not only will take care of that myopia, but they'll also take care of loss of myopic condition over there and so on. And that's one kind of glasses that's being used, which is really not that easy. It's also not comfortable. It's also ugly looking. So one needs to worry about what could be the kind of mechanics, the kind of polymer you might want to use, the kind of lenses that you might actually want to grind in a manner that focusing happens not just at the foveal point, but also regions surrounding it. This is a challenge that, again, ophthalmology is not able to address. And it's not a biological problem, it's really a problem of engineering. So, this is a very interesting, a professor of physics from Oxford has come out with this idea. And that is, even today, if you look at in India, 10 million people are blind. By blind, I mean 360. What you see at 60 feet, they can't see until they cut 3 feet and so on. Legally blind. Out of these 10 million, about 7 million are blind because they have not had a cataract operation. 1 million are blind. 1/10 of Indian blindness is due to uncorrected refractive errors. In other words, they don't wear glasses or contact lenses. Oftentimes they are given glasses, and once the glass breaks, they no longer have the ability to go and buy another pair of glasses, and they just decide this is destiny or fate. Is it therefore possible that one can even try and use methods by which you can actually have lenses or eyeglasses that I can change according to whatever power I need? Can it be one that is essentially unbreakable? George Silver of Oxford decided he will try and do that. And he said one way to do that is completely remove the glass, the hard material. I would rather use a silicone bag, transparent silicone bag or mylar bag, whatever, into which I'm actually going to put liquid. And the liquid he's using is a silicone oil, refractive index approximately the same, anywhere from around 1.35 to 1.5, and you fill it the way you want. It's a self-fillable. That's how it originally looked. And it's been tried to about 45,000 people in Ghana. So this gentleman here said, this glass is not good enough for me. You can actually put in a little bit more of silicone oil in there and correct his glasses till he finds comfort. It has now moved on into a little bit more jazzy looking thing. Here is such a simple idea of removing hard material, putting in essentially a refractive material that is again flexible as a liquid and so on. Whether any improvement can be made of these is another point of interest. What about a cornea? Millions across the world need corneal replacement, and the number of cornea available is far less than one would imagine. So the question is, can we therefore make an artificial cornea? If I can make that, it must be compatible with the surrounding tissue, with the underlying tissue. The surrounding tissue is the conjunctival membrane. Below that is the aqueous humor. So I shouldn't have inflammation, infection and so on. It must be transplantable, removable if you would like to. It must have acceptable refractive indices, flexible, non-abrasive, long-lasting, hydrated and oxygen permeable. In other words, it must be a cornea. This lady here, Professor May Griffith, from the Ottawa Health Research Institution in Canada, has actually tried using recombinant human collagen sheets. She's also tried using polyl-lysine, glycolic acid copolymer. But since then, whole variety of polymers have been used here in a manner that they will actually be biocompatible. They can be put in there. They will not be abrasive, because every time you blink, you will actually lose a little bit of the corneal epithelial cells in the natural eye. But because there are stem cells in there, they keep on making themselves. But in this case, if we're going to put essentially a an artificial insert in there, it must be so smooth, non-abrasive, that I do not lose my focal vision and so on. So what's the best one? We still do not know. We have been trying. May Griffith's polymer, which she uses recombinant human collagen, and to this day it looks good on animals. We don't know how it is going to be on humans. The problem therefore is going to be one of the mechanical properties, the transparency, the elasticity, and clearly how rugged this is going to be. But if you do this, I think there will be tremendous hope that will come. In India alone, for example, we need something like about 140,000 donor cornea every year. We do not harvest more than about 4,000 or 5,000 at best. So it's very clear that we need such material. Turn now to the vitreous humor, which is the back of the eye. This is one of the major difficulties that we have. It's also going to be a major difficulty in India simply because of diabetes increasing. This is a transparent gel. On the front of it is the lens which is kept in the capsule. On the back of it is the retina, and it holds the two like so. Now, with age and with time, the vitreous gel, the gel essentially is made of chondroitin sulfate, hyaluronic acid, and collagen. It's a biopolymer complex which is completely transparent. Therefore, it doesn't in any way affect vision or the focal length. It's a 100% transmitted situation. With time, the gel actually liquefies. On the one hand, it is good, on the other hand, it's not very good. Once it separates from the retinal surface, in other words, the posterior vitreous detachment, then if the retina is well connected with the choroid around, you have no difficulty. Therefore, in one way it is good, since such a detachment decreases the chance of what people refer to as proliferative diabetic retinopathy. Retinopathy is one where more and more blood vessels keep on forming on the retina. When such blood vessels form, chances of blood vessels bursting is very distinctly possible, and that would of course affect vision. One of the major difficulties of diabetic retinopathy, or diabetes itself, is this thing called diabetic retinopathy, which can cause hemorrhage, and therefore have this difficulty. Fortunately, in the case of PVD, since the attachment is gone, the possibility of retinal detachment and retinopathy is somewhat restricted. In diabetics, we would therefore like to hasten PVD if possible, so that retinopathy doesn't happen, and no peeling-based hemorrhage will happen. As of today, such a thing is actually done by using a bunch of enzymes, hyaluronidase and collagenase and so forth. And there is now a possibility of a product called Vitreosol, which we are currently testing out at LV Prasad. And you can find that in proliferative things, you can actually go from that situation to a little better situation, but we do not know if that's the best. What would therefore be a way of actually attempting to do such PVD hastening is another question that we are raising. Finally, if you turn to the retina, while biology is trying to address retinal disorders, notice compared to the cornea, you've got a difficulty here. It's a 10-layer tissue, and in that 10 layer, you've got at least 6 or 7 different kinds of cell types. So, biologically remaking a retina is going to be a major challenge indeed. And here is where electronics has taken on a rather hopeful possibility. And that is something that we should talk about, but even before that, cancers of the eye, in particular cancers of the retinal region, called retinoblastoma, is seen quite rampant in it's essentially a children-based thing. More often than not because it's genetic, but you can also have sporadic situations here. And the way to do that essentially is to do surgery and also remove all the cancer tissue by using anti-cancer drugs like cisplatin, carboplatin, and so on. The problem there is how do you reach the retina? You can go periocular, Ideally, if you can really go for the top using eye drops or something like that, top subconjunctival, that will be a terrific way of doing that. There seems to be one way of doing that, and we have done this on animal experimentation in 2007 and 2008. Dr. Honavar, my colleague who has been doing this, has now been able to put this carboplatin molecule in, we have used PAMAM, the dendrimum, carboxylated dendrimum, this is an amino side chain, but carboxylated one also. And if you encapsulate it and you put it in there, still subconjunctival or periocular, are there better ways of doing that? The local concentration of carboplatin that goes on to the tissue over there is at least 100-fold higher if you were to use a nanoparticle encapsulation. But are there better delivery vehicles, something we need to certainly think about? That again, we would require nanoencapsulation situation. Here is a remarkable situation where you actually put in, rather than the damaged retina, can I really put in there, let me go over here. Rather than using the usual biochemical approaches or biological approaches, what Professor Humayun has done is actually put in electronic circuitry. He's actually put in there a whole bunch of pixels which light activated electrical circuitry goes on. The question there was, of course, how many pixels would be good? The resolution would by and large depend on how this is going to be. See, here is how the thing works. He has put in the implant over there, and that contains a sheet that has, let's say, 16 pixels, 50 pixels, whatever you want. And you put in a pair of glasses here, and the glasses see the image. The signals are sent to the handheld device, process information sent back to the glasses. Okay, so you really have to have not only the implant over there, but you also have glasses that actually essentially will bring you the light into the implant, and you have a handheld device like a mobile phone or something like that. So you end up getting information that actually stimulate the retina, and the stimulated retina sends information to the brain, and there is partial visual recovery. And Professor Humayun was trying to come up something like about 12 patients now says, even with 16 pixels electrodes, how much our first 6 subjects have been able to do was quite a lot. The pixel number will have to increase upon experience. Currently 75 subjects have volunteered for this trial, and the number that he has put in there is something like about 256 pixels. Would that become better? Is there a better methodology than just using pixels of this kind? We do not know. There is even a question, can there be a contact lens retinal implant? The reason I am saying that is because just last Monday's Economist talks about this gentleman called Babak Parvez. I know very little about it other than just reading about that. What he has done, he has taken a regular contact lens in which he has incorporated electronic circuitry, including LEDs for on-eye displays, radio or wireless communication that can go on, an antenna for collecting power so I can regenerate myself from a cell phone or a mobile, something of that type. And what it essentially does is, because of this, it's able to, you can use it essentially as a GPS or navigatory aid and so on. If you can do that, essentially putting in there, now the amount, what you can put in there is hardly a micron or so in diameter, therefore it does not affect visual function. On the other hand, it also adds on this little circuitry that gives you navigational aid and so on. If you can do that, is it possible for us to actually go right on into collecting minus the retina, essentially only the information that goes directly to the brain? That's a question one needs to ask. There are even more daring questions I will leave you with. Is it possible to deliver genes using nanomaterials? It's not an easy question, because the DNA molecule itself is multi-nanometer in dimensions in terms of length and so on. Is it even possible to think of it? Because that's one of the easiest approaches to approach the eye. Is it possible, therefore, to use eye drops that can reach the retina? notice the impediments. When you want to put an eye drop over there, you have the tear film, you have the cornea, then you have the aqueous humor, then you have the iris, then you have the lens, vitreous humor, finally you go to the retina. Is it possible at all to even think of an eye drop that will go directly to them? If you do that, the tremendous market for it, both for glaucoma patients and for retinal patients. Can I use the tear fluid as a diagnostic material? Today's biology, including proteomics and any other metabolomics that we have possibilities, is it now possible for me to look at the tear fluid, use it as a diagnostic material, at least for certain kinds of diseases or disorders? We do not know the answer to this question. Some people even thought about re-engineering taste bud cells, which have exactly the same kind of a complexity that a retinal tissue has with respect to circuitry, intracellular trafficking, and getting on into neurons and so on. Last, can I transplant an entire donor eye? Suppose I remove the entire eyeball. Is it possible now to take a donor eye? What's the difficulty? Difficulty is going to be in terms of the connectivities with the neural, with the brain. If I can take a broken hand, a chopped off hand, it is possible now for a plastic surgeon to put an entire hand back. And you have exactly the same height, maybe a little less in terms of complexity, but if you already have successful ways of a plastic surgeon taking a chopped off hand, putting it back, stitching it back together, and ask the patient actually to recover pretty much his original hand, is it too difficult to ask a question of transplanting an entire donor eye? I leave you with questions of this kind. Some of these are clearly possible in the next 2, 3, 4 years. For example, eye drop reaching the retina, I think is no longer a great difficulty. Gene delivery, is nanomaterials the best way, or alternate possibilities? Can I use a tear fluid as a diagnostic material? I think this also is a possibility that people can work on. I do not know about how easy re-engineering a taste bud to repair the retina. I haven't talked about our stem cell work that you've been looking forward to, Professor Wegner, but it is possible now to take oral mucosa cells, make the corneal epithelial sheet from that, and put it on the eye in order to do corneal damage repair. The difficulty there, of course, is there are blood cells in the oral mucosa. There are no blood cells in the cornea. She had to address that problem. Maybe one can use inhibitors like VEGF receptors and so on. But is it possible to consider taste bud cells and go on to produce other kinds of tissues and transplant an entire cornea? I leave you more with these questions than any answers. Thank you.